Bismillahirrahmanirrahim. Assalamu alaikum, dear students. I am Asma Kamran. I hope everyone is enjoying a good health. In our previous lecture, we learned peroxisomes, glyoxysomes, vacuoles, mitochondria, plastids, structure and functions of chloroplast and centrioles. Our today's objective is cytoskeleton, cilia and flagella, structure of cilia and flagella, nucleus, prokaryotic and eukaryotic cells, and in the end exercise. First topic is cytoskeleton. Cyto means cell, skeleton the structure which provide support and maintain shape of the cell. In case of cell, there are three elements which make up cytoskeleton. They are present in the cytoplasm and distributed from nucleus to the plasma membrane. These three elements are microfilaments, microtubules, and intermediate filaments. As you can see in this figure, they are in the form of network, they are in the form of long bundles, or in the form of filaments. These thin filaments are actin filaments or microfilaments. Thick one are microtubules, and these are intermediate filaments. As the name shows, they are intermediate in size between the microfilaments and microtubules. Now the first one is microfilaments. Microfilaments are made up of a protein that is called actin. That's why they are also known as actin filaments. They are very thin, of about 7 nanometer in diameter. They are made up of two types of actin, G-actin and F-actin. G-actin means globular actin, which are globular structures. G-actins are arranged together to make up one long filament, or F-actin or fibrous actin. Two F-actin, they twist together to make up one microfilament. As you can see in this figure, these rounded structures are G-actin or globular actin. They combine together to make up F-actin, and two F-actin twist together to make up microfilament. Microfilaments in many cells, they also have another protein that is called tropomyosin. Tropomyosin is also made up of two chains, and these two chains of tropomyosin twist around the actin filament. And there's another protein or a third protein that is called troponin. Troponin consists of three subunits which are arranged in the form of a triplet, as you can see in this figure. And troponin is attached to tropomyosin at certain intervals. Together, actin, tropomyosin and troponin, they help in contraction, in cyclosis, and also to provide shape and support to the cells, especially to the plasma membrane. Microfilaments are present just under the plasma membrane, so they provide support to the plasma membrane. Microfilaments also help in the circular streaming movement or cyclosis. Remember, we had seen that in cytoplasm, the cytoplasm has the ability to move, so that due to which organelles, they also move from one place to the other, and other components in the cytoplasm, they can move. This is a figure that shows all three types of protein. G protein, these are the globular structures, and these globular or G-actins combine together to make up one long chain, that is F-actin. So two F-actin twist around each other to make up one microfilament. Second protein, tropomyosin, is also in the form of long chains. So two chains of tropomyosin, they twist around the actin or microfilament. And the third protein, troponin, in the form of three subunits or triplets, is attached to tropomyosin at certain intervals. So together they help in contraction and in providing support to the cell. Microfilaments are abundant in many cells, especially in the muscle cells, in the skeletal muscles, where they form bundles. These bundles are called microfibrils. And in this figure, this is one muscle fiber with number of myofibrils. And these myofibrils, they have actin filaments in it. In this figure, these red lines, these are representing the actin filaments. Together with myosin filaments, they help in the contraction of the muscle. Microfilaments in the cell usually are present in the form of bundles, or they make network in the cytoplasm. This is another figure that shows amoeboid movement. This is unicellular microscopic organism, amoeba. In this case, you can see this is cytosol in the center. Remember, cytoplasm, the central part of cytoplasm is less viscous, so it is called cytosol. And the outer part of the cytoplasm is more viscous, that is cytogel. In this figure, you can see that cytogel have network arrangement of microfilaments, and cytosol have subunits of actin filaments or microfilaments. So these subunits of actin filament plus sol nature of the cytoplasm helps in the movement or in the flow of cytoplasm to a finger-like structure that is called pseudopodium, which help in the locomotion of amoeba. So this movement is called amoeboid movement. The second figure is representing the cyclosis or streaming movement of cytoplasm. This is the central part of the cytoplasm, which is cytosol. Here these actin filaments are arranged in parallel chains, but in the cytogel you can see that actin filaments are in the form of network, and these filaments, which are in rows in case of cytosol, they help in the movement of the cytoplasm, and this movement is called cyclosis. And in this case, you can see that due to the cyclosis, chloroplasts can move from one place to the other place. Especially they move to the site where they can absorb maximum sunlight. Now, second one is microtubules. Microtubules are thicker and they are hollow from inside. They are in the form of long cylinders, which are of about 25 nanometer in diameter and 0.2 micrometer in length. They are made up of another protein that is called tubulin. Tubulin is made up of two subunits, alpha subunit and beta subunit. In this figure, alpha unit is represented in this red color and beta is in blue color. And when they arrange together, one tubulin molecule is arranged with the second tubulin molecule in such a way that alpha tubulin of one tubulin always come opposite to the beta of another molecule. So they arrange to form paired filaments, linear filaments in long rows, and these filaments twist to make up a tubule, that is microtubule, which have a space in the center. And this is a cross section in which you can see that alpha and beta, they alternate each other with a central space. Microtubules in case of plant cells are present, freely dispersed in the cytoplasm, and microtubules also help in the formation of mitotic apparatus. Mitotic apparatus is that which helps in the attachment of chromosomes and then in the movement or distribution of chromosomes to the daughter cells. In case of animal cells, centrioles are present. Centrioles helps in the formation of this mitotic apparatus with the microtubules. But in case of plant cells, centrioles are absent. So freely dispersed microtubules, when the cell is about to divide, they arrange themselves to make mitotic apparatus, then help in the distribution of chromosomes. And microtubules, they are also involved in the formation of centrioles. Remember, centrioles are made up of triplets which are arranged at the periphery. These triplets are made up of microtubules. Cilia and flagella, which are the organs used for locomotion in case of some cells and organisms. Cilia and flagella are also made up of microtubules. Cilia and flagella, they arise from basal bodies. Basal bodies have a structure just like centrioles, so they are also made up of microtubules. In these figures, you see these are different types of bacterial cells. They have flagellum, and this flagellum helps the bacterial cell to move. And this is a microscopic paramecium with number of cilia, which help in locomotion. Now the third one is intermediate filament. As the name shows, they are intermediate in size between the microfilaments and the microtubules. Intermediate filaments are 8 to 10 nanometer in diameter, and they are made up of a protein that is called vimentin. Some of the intermediate filaments are also made up of keratin proteins. And just like microfilaments, these vimentin subunits, they are arranged in the form of long chains or strings to make up long chains of intermediate filaments without any space or without any lumen. So they are like microfilaments but thicker than the microfilaments. The function of the intermediate filament is to make up a network which is present in the cytoplasm, extend from the nuclear envelope up to the plasma membrane. As you can see in this figure, that intermediate filaments, they make a basket-like structure around the nuclear envelope. Here the nucleus is not shown, only the intermediate filaments are shown here. And in the cytoplasm, they make this network, and just beneath the plasma membrane, again arranged in parallel manner. So they provide support to the plasma membrane as well as to the nuclear membrane. This table is showing a comparison between microtubules, microfilaments, and intermediate filaments. You see, microtubules are hollow tubes with 13 columns of tubulin molecules, and microfilaments are made up of 2 twisted strands of actin. Same is the case with intermediate filaments. These are made in the form of long fibers or in the form of thick cables. And microtubules are 25 nanometer with a central space or lumen which is of 15 nanometer. Microfilaments are of 7 nanometer. Intermediate filaments are of 8 to 12 nanometer. Microtubules are made up of tubulin. Each tubulin is made up of alpha tubulin and beta tubulin. And microfilaments are made up of actin, and intermediate filaments are made up of vimentin or keratin proteins. And microtubules, they help to maintain the shape of the cell, help in the motility or in the movement of the cell if they are present in the form of cilia and flagellum. And they also help in the formation of mitotic apparatus to help in the movement of chromosomes during cell division, and they also help in the movement of organelles. Microfilaments, they also maintain shape, help the cell to change its shape if they are present in the form of bundles in the muscles, help in the contraction of muscles, and responsible for cytoplasmic streaming, and also the movement of cell as in the case of pseudopodia, and they also help in cell division. They form a cleavage. When the nucleus divides, the cytoplasm divides by a cleavage formation. This cleavage or furrow is formed due to microfilaments. Intermediate filaments, they also maintain shape and helps to provide mechanical support to the nucleus and plasma membrane. Also form a network just inside the nuclear membrane. This is called nuclear lamina. Its function is to provide support to the nuclear envelope or nuclear membrane.
The cytoskeleton is a dynamic network of filamentous proteins that extends throughout the cytoplasm, forming the struts, cables, and girders that give the cell its shape, internal organization, and mechanical support for movement. There are several types of protein filament, each built from subunits that rapidly come together or break apart, allowing these elaborate structures to be assembled and dismantled wherever they are required by the cell. Microtubules are stiff, hollow tubes that anchor other organelles and serve as tracks that guide the movement of vesicles and other cell components. Actin are thin, flexible filaments that form cross-linked bundles and branching networks. Actin is particularly important for cell movement. They are central to the contractile engine in muscle cells and provide the mechanical force for cell movement at the plasma membrane.
Now this is another table that is showing a comparison between microfilaments, intermediate filaments, and microtubules. It is slightly blurred, but I think this will be clear to everyone. What are the subunits or the proteins that make up actin filaments, intermediate filaments, and microtubules, and what are the functions of these cytoskeleton? Now the next topic is cilia and flagellum. These are different from each other as cilia is smaller and flagellum are longer in size. Cilia is from a Latin word cilium, that means eyelash or hair. Flagellum is from a Latin word flagella, means whip. Cilia and flagella both are hair-like projections which are extending or present on the outer surface of the cell. Cilia and flagella, their internal structure is same, but they are different in size, number, and the pattern of movement. Cilia are smaller in size and the flagellum are longer in size. Flagellum are present in less number and cilia are present in numerous or more in number. Their internal structure is same. Flagella, they are few in number, as I told you. Mostly in many cases they are present only one, or they may be two or four, and they show undulating movement or a spiral movement which starts from the base of the flagellum and up to the tip of the flagellum. And if they are present in a pair, that is two or maybe four, each and every flagellum moves or it beats independently. As in these figures, this one is showing a spiral movement which starts from the base and moves up to the tip, and this then helps in the forward movement of the cell or organism. Same is the case over here. This is a spiral movement and helps in the movement of organisms, and this movement is undulating movement. Cilia are short, more in number. Usually whole of the surface of the cell or organism is covered by cilia, and they beat in a perpendicular manner. And the beating of cilia is of two types, metachronous and synchronous. Metachronous is that where the cilia of a row, they beat one after the other, as you can see over here. First of all, one cilia bends, then second, then third, then fourth, then fifth, and so on. And then after bending, first cilia straighten up. This is followed by the second, third, fourth, and fifth. So this type of movement is called metachronous movement or metachronous rhythm. And the second one is synchronous, where all the cilia on a row, they beat at the same time or simultaneously. This is called synchronous. As you can see in this figure, this is a surface on which these cilia are present. You see all the cilia, they are beating at the same time, or they show movement at the same time. But in this case, one cilium show independent movement or independent of the other or the neighboring cilia, so that one is followed by the second, then third, fourth, and so on. So they move one by one. Now the structure of cilia and flagella. Cilia and flagella, they consist of two parts. The basal body, which is present beneath the plasma membrane. And the structure of the basal body is just like a centriole, which consists of triplets of 9 tubules. And each triplet consists of 3 microtubules. And a part which extends out of the surface of the cell, this long part is called exoneme. Exoneme is covered by cytoplasm and a plasma membrane, which is continuous with the plasma membrane or the cell membrane of the organism. Exoneme consists of doublets of microtubule, and the structure of exoneme of both cilia and flagellum is same. Now, what is the structure of exoneme? Exoneme consists of 11 long microfibrils or microtubules. Out of these 11, 9 are arranged in the periphery in the form of a double structure, and 2 single tubules are present in the center. As you can see in this figure, these 9 microtubules are arranged at the periphery, and these 2 single microtubules are in the center. These 2 single microtubules are covered by a sheath, that is inner sheath or a central sheath, and these 2 microtubules are connected to each other by this central bridge. This arrangement of 9 filaments in the periphery and 2 in the center, this is called 9+2 pattern, and this helps in the movement of cilia and flagellum. Each peripheral microfibril or microtubule is made up of 2 subfibres, so that is called a doublet. As you can see in this figure, each doublet consists of 2 microtubules. One microtubule or one subfiber is complete and other one is C-shaped or incomplete. This complete one is called A tubule and C-shaped or incomplete is called B tubule. To the A tubule or to the complete subfiber, 2 arms are attached which are made up of dynein protein. This is outer dynein and this one is inner dynein. Each doublet is attached to the central tubules with the help of radial spokes, and these radial spokes, they extend from A tubule to the central sheath. Remember the central sheath that covered the 2 central microtubules. And each doublet is also connected with the neighboring doublet by another protein. This is called nexin. So exoneme have 3 types of cross-linkage: nexin, radial spokes, and the bridge which is present between the central tubules. Basal bodies are also present at the base of cilia and flagella, and their structure is just like the centrioles, made up of triplets of microtubules. And from these basal body, exoneme arises, which make up cilia and flagella. Now, what is the mechanism of bending? How these microtubules help in the movement or in bending of cilia and flagella? The dynein arms of one doublet attach to an adjacent doublet and pull it so that they slide in opposite direction. When the dynein arm attached to the second doublet, it pulls that doublet and then release it, then again attach to the doublet but to another site, to another place. So it pulls it in opposite direction. So same is the case in bending. So this is just like the walking of the dynein arms. So dynein arms are responsible for the bending or for the movement of cilia and flagella. Dynein actually consists of enzyme which hydrolyzes ATP. ATP, when hydrolyzed, it produces energy. Remember, ATP is broken down to release energy, and this ATP then activates or stimulates the motor part of dynein, and dynein then starts walking on the adjacent microtubule doublet and helps in the bending of the microtubule, or microtubules, they slide over each other. But the sliding is not very far, or it is restricted due to the central bridge which is present between the 2 central tubules, and also due to radial spokes and the nexins which are connecting the 2 doublets with each other. So when the microtubules or when the cilium and flagellum bend, the bending is only to some extent because it is restricted due to the cross linkages present in the flagellum and cilium or in the exoneme. And these cross linkages are bridge which is present between the central tubules, radial spokes, and nexen. This is another figure that shows the Dynein arms. This is one doublet. This is A tubule. This one is B tubule of this doublet. This is second doublet with A and B tubule. You see the Dynein arms which are present at the A tubule of one doublet. They attach to the B tubules of the second or the neighboring doublet, and then they pull it. So they help in the bending of these doublets, and when they bend, cilia or flagellum bend. This is another figure. In this figure you can see that Dynein arms, they just look like two arms or legs which help in the walking on the second doublet. So they actually move just like the two limbs, or just like a cat climbing on a wall. So help in the sliding of two doublets against each other, and then in the bending of the microtubules of the doublet.
Motile cilia and flagella are hair-like appendages of cells that generate fluid flow along tissues or allow cells to move through their environment. To better understand how these appendages generate motility, we flash froze sea urchin sperm cells while their flagella were actively beating, and then directly visualized the molecular machines inside the flagella through cryo-electron tomography. Thousands of motor proteins, called dyneins, are arranged regularly along the microtubule scaffold. Through classification, we identified several different functional states of the dyneins as they step along the microtubules. We demonstrate a detailed stepping mechanism. From the relaxed post-power stroke state, the dyneins release from the microtubule, swing forward, and rebind to the microtubule. During the power stroke, the cargo is pulled forward by the action of dynein. As long as ATP is present, dyneins continue to walk along the microtubule. However, if dyneins receive inhibitory signals, they can be kept in intermediate states that resemble the post-power stroke state, leaving them unable to generate motion. Our direct visualization of specific dynein conformations on either side of the flagella suggests a switch inhibition mechanism generates the flagellar beat. As one side is inhibited, the other side is active. In our model, regulatory signals result in inhibited dyneins on one side. This allows dyneins on the opposite side of the flagellum to walk along the microtubules. Molecular linkers resist this sliding, which causes the flagellum to bend in one direction. By periodically switching the side of dynein inhibition, the flagellum alternates the direction of bending, resulting in the typical whip-like motion. The propagation of dynein inhibition in different regions of the flagellar wave along the length of the flagellum leads to the characteristic beating motion. The dynein inhibition may occur in an all-or-none fashion, similar to an action potential, which could explain how cilia and flagella can switch their bending direction back and forth in less than 1/100th of a second and continue beating for days.
Now the movement of cilia. In 1955, it was suggested that cilia, its movement or bending is in the contraction of double fibrils in two groups, which contract one after the other. Out of 9 double fibrils which are present in the periphery, 5 contract at the same time or simultaneously. So cilium bends to that site where these 5 doublets are bending or contracting. When they are contracting, cause the bending, this is called effective stroke. And when these 5 doublets, they relax, the remaining 4, they start contraction, due to which the cilium, it becomes straight. This is called recovery stroke. as you can see in this figure. In the effective stroke, the cilium, it is bending, and in the recovery stroke, the cilium is now starting to stand straight or erect or straighten up. That is called recovery stroke. In the effective stroke, out of 9 doublets, 5 doublets they contract simultaneously, and the 5 doublets, when they contract, the cilium bends to that side where these 5 doublets are present, and the remaining 4 are relaxing at that time. So when these 5 they start relaxing, the remaining 4 start contraction, due to which the cilium straightens up, and the first one is called effective stroke and the second one is recovery stroke. And the flagellum, as I told you before, that its movement is called undulating movement, and its movement is the movement which starts from the base of the flagellum up to the tip, and this then helps to move or to propel the cell or organism in a forward direction. Now the next and the last or the third major and the prominent component of the cell is nucleus. It is present in the center in case of animal cells, but remember in the plants, due to the central vacuole, the nucleus is pushed towards one side. And nucleus consists of nuclear envelope or nuclear membrane, nucleoplasm, which is just like cytoplasm, but slightly different in chemical composition from the cytoplasm. It have more nucleic acids in it, that's why it is called nucleoplasm, as it is present in the nucleus. It have nucleolus, which is a part inside the nucleus and is responsible for the formation of ribosomes. And it also have chromatin. Chromatin is a network which condenses to make up chromosomes. Now the first part of nucleus is nuclear envelope, which is also called nuclear membrane. Nuclear envelope, just like the membrane of mitochondria and chloroplast, is a double membrane. Outer nuclear membrane and inner nuclear membrane. There is a space between the outer nuclear membrane and the inner nuclear membrane. This space is called perinuclear space, and it is filled with a fluid. Nuclear membrane, just like the plasma membrane or cell membrane, is made up of lipid bilayer and proteins. The outer membrane of the nucleus or outer nuclear membrane is covered with ribosomes, and outer nuclear membrane is also connected or attached to the rough endoplasmic reticulum. And another important feature of the nuclear envelope are the pores or the nuclear pores. These nuclear pores consist of specialized transport proteins which are called nucleoporin. Remember, porin are also present in the membranes of mitochondria and chloroplast, but as here these are present in the nucleus, help in the movement of molecules and ions from cytoplasm to the nucleus and from nucleus to the cytoplasm, so they are called nucleoporin. This is a figure that shows all these features. This is nuclear envelope made up of 2 membranes, outer nuclear envelope and inner nuclear envelope, with a space in between that is called perinuclear space, and these are the nuclear pores. This is nucleolus, that is chromatin, and you can see that outer nuclear membrane is connected to this rough endoplasmic reticulum, and on the surface of outer nuclear envelope ribosomes are also present. This is another figure, magnified figure of a part of a nuclear envelope. This one is outer nuclear envelope, this is inner nuclear envelope, and here the perinuclear space is very much clear. And these circular structures, these are the nuclear pores, and these are ribosomes which are attached to the outer nuclear membrane. Now the nuclear pore. Nuclear pore are actually not the pores or the holes. Actually they consist of a complex proteins that make up a complex structure. So This nuclear pore help in the exchange between the cytoplasm and the nucleus, and from nucleus to the cytoplasm. And the exchange usually includes RNA, ribosomal proteins. RNA is especially the ribosomal RNA, messenger RNA, ribosomal RNA, and ribosomal proteins are synthesized in the nucleolus. So they make up ribosomes, which are then transported from nucleus to the cytoplasm. Same is the case with messenger RNA, which is synthesized from DNA, and then it is transported from the nucleus to the cytoplasm, where they attach to the ribosomes and then translate protein or synthesize protein. And some enzymes which are synthesized in the cytoplasm, like DNA polymerase. This is an enzyme which help in the duplication of DNA. Some carbohydrates, lipids, they also move from cytoplasm to the nucleus. Small molecules, they can simply diffuse from outside to the inside of the nucleus, or maybe from inside the nucleus to the outside to the cytoplasm. But the large molecules, they require specific or special signal sequence to move in and out. This sequence is identified by the nuclear pores or the proteins which make up the nuclear pore complex and helps in the transport of these large molecules from nucleus to the cytoplasm or from cytoplasm to the nucleus. Then inside the inner nuclear membrane, there is a network of intermediate filaments that is called nuclear lamina. As you can see in this figure, this network present just inside the nuclear membrane. This is made up of intermediate filaments, and this is called nuclear lamina. Its function is to maintain the shape of the nucleus by providing a support to the nuclear envelope. Third component is nucleoplasm. Nucleoplasm is a cytoplasm present inside the nucleus, but it is different from the cytoplasm because its chemical composition is slightly different from the cytoplasm. It has some specific proteins, enzymes like DNA and RNA polymerase. DNA polymerase is for duplication of DNA. RNA polymerase is for the formation of different RNA, especially the messenger RNA. Free nucleotides are present in the nucleoplasm. These free nucleotides are used up for making new DNA strands. Some metal ions like magnesium for the synthesis of DNA and RNA. And nucleoplasm also consists of proteins, especially the histone proteins and non-histone proteins. Histone proteins together with DNA make the structure of chromosome, that typical structure of chromosome. Another component which is present inside the nucleus, it is called nucleolus. If they are more in number, they are called nucleoli. And nucleolus, they are not bounded by any membrane, and they may be a single or one nucleolus inside the nucleus in many cells, or there are many cells which have more than one nucleolus inside the nucleus. And nucleolus usually appears during the interphase and disappears during cell division. Interphase is a time period between the two cell divisions. Interphase is the time when the cell is growing and increasing in size and also preparing itself for the next division. So this is a time of rapid metabolic activity because cell is growing and increasing, it is developing, and it is also preparing itself for this next cell division. At this time, nucleolus is prominent, or it appears in the cell, or it is present in the cell. But when cell division takes place, at that time nuclear membrane also dissolves and the chromatin network condenses to form chromosomes. So nucleolus disappear at the time of cell division. Nucleolus consists of two prominent areas. The outer one is a peripheral area which have granules, so it is granular area, and this granular area consists of ribosomal subunits, larger ribosomal unit and smaller ribosomal subunit, while the central part of the nucleolus have fiber-like particles or structures in it. Mostly these are the rRNA and rDNA, or ribosomal RNA and ribosomal DNA. Ribosomal RNA make up ribosomes, so it is the part of ribosomes, and ribosomal DNA is responsible for the synthesis of that proteins which then make up ribosomes. So nucleolus, their function is the formation of ribosomes. Now another major component of nucleus is chromatin. Chromatin are thread-like structures which are present in the form of a network in the nucleoplasm, and chromatin are made up of DNA and proteins, histone proteins as well as non-histone proteins. During cell division, this chromatin network, it condenses to make up chromosomes, and chromosomes are made up of DNA and proteins. Chromosomes have genes on it, so these chromosomes then transport these genes from one cell to the other cell. Chromosomes are the structures which can also be seen with the light microscope, and single chromosome consists of two parts which are called chromatid. Chromatid are attached to each other at a point which is called centromere. Centromere is present in a thin segment which is thin than the rest of the chromatid. So this thin segment is called primary constriction. And each chromatid towards its outer side at the primary constriction or at the centromere has a protein called kinetochore for the attachment of microtubules during cell division. As you can see in this figure, these are two chromatids which are connected to each other at centromere. And in this figure, you see two chromatids which are connected to each other at centromere. Centromere is present in a constriction which is called primary constriction, and towards the outer side of the centromere, protein kinetochore is present on each chromatid, and to these kinetochore, microtubules of the mitotic apparatus, they are attached. These red filaments are representing the microtubules of the mitotic apparatus. And these microtubules then help in the separation of these two chromatids so that each chromatid move to one cell and other to the other cell. And some of the chromosomes, not all chromosomes, have another constriction or constriction other than this primary constriction. This second constriction is called secondary constriction, or it is also called nucleolar organizer, because this constriction have the genes or a DNA which is responsible for the formation of nucleolus. And some of the chromosomes, not all chromosomes, just adjacent to the secondary constriction have a knob-like structure which is called satellite. It is believed that satellite consists of useless or junk DNA which is of no use. And the secondary constriction and satellite, it is not present in all chromosomes. Only some of the chromosomes have these structures. And the terminal ends of the chromosomes are called telomere. Telomere prevents the two chromosomes to attach at their ends, and chromosomes are stained with specific stains or with specific colors, that is acetocarmine or acetoarsine. Acetocarmine is a red stain and acetoarsine is purple red stain. And when the nucleus is stained with these dye or with these stains, some of the areas of the chromatin, they take lighter stain, and some areas they take more stain. The areas which take light stain, they appear lighter in the stained nucleus, while the areas that take up more stain, they appear darker. The areas which appears lighter, they are called euchromatin, and the areas which appear darker, they are called heterochromatin. It is believed that euchromatin is a true chromatin. Eu means true, or here the chromatin material is loosely packed. The genes or the DNA which is present here in the euchromatin, it expresses itself, while in case of heterochromatin, the chromatin material is very tightly packed. This is the area where the genes, they very rarely express themselves. And this is a figure that is showing the karyotype of human. Karyotype means all the chromosomes, their size, structure, shape. You can see that each and every chromosome is different from each other. There are 23 sets or 23 pairs of chromosomes, but each and every chromosome is different from each other. So chromosomes are identified on the basis of their size and shape.
Eukaryotic cells, which are complex cells that have organelles, genetic information is stored in a structure called the nucleus. Known as the control center of the cell, the nucleus is contained by a double membrane called the nuclear envelope, which works to protect the DNA by controlling what types of molecules can enter or exit. The nuclear envelope uses pores that allow certain substances to enter and leave. Inside the nucleus is an area called the nucleolus. The nucleolus plays an important role in turning genetic information into reality. RNA is synthesized in the nucleolus from the DNA code that makes up who you are. Parts of ribosomes, a cell organelle, are made in the nucleolus and moved through pores in the nuclear envelope. A new piece of furniture is usually carried through a doorway as a box full of materials before it's assembled inside a house. That's the way that the pores in the nuclear envelope work with ribosomes. Once the subunits of ribosomes are transported outside the nuclear envelope, they can be assembled. Ribosomes are very important to living things. They're the site where proteins are made. Proteins are used in the body to build structures. In the same way that lumber is used to build a house, proteins are the scaffolding of the human body. When cells divide, they need to pass genetic information onto new cells. The genetic information in the nucleus is stored in long strings of DNA called chromatin. When cells prepare to divide, they organize their DNA. Chromatin is condensed down into structures called chromosomes. Chromosomes are split and passed on to offspring in order to pass a copy of DNA to newly formed cells. Possibly the most important part of cells, their DNA, is protected and stored in the nucleus.
Now the last topic of this chapter, prokaryotic cell and eukaryotic cell. As you can see in a figure, prokaryotic cells are usually single-celled and eukaryotic cells are single-celled as well as they make up multicellular organisms. Prokaryotic cells, they make up organisms which are called prokaryotes, and eukaryotic cells make up organisms which are called eukaryotes. This table is showing a difference between prokaryotes and eukaryotes. Prokaryotes usually include bacteria, archaebacteria, and also cyanobacteria or blue-green algae. Eukaryotes are fungi, protozoa means microscopic organisms, algae, and all higher multicellular organisms including plants and animals. Prokaryotes, they lack a nucleus, consist of only one circular chromosome which is not bounded by any nuclear membrane. And eukaryotes, they have true membrane bounded nucleus and which consist of number of chromosomes. In case of prokaryotes, there are no histone proteins, but eukaryotes have histone proteins. Prokaryotes have only organelle, which is ribosome. Remember, that is smaller as compared to eukaryotic ribosome. It is 70S ribosome, and eukaryotes have 80S ribosomes. Prokaryotes, they do not have any membrane-bounded organelle, but eukaryotes have membrane-bounded organelles like mitochondria, endoplasmic reticulum, Golgi apparatus, etc. Prokaryotes and eukaryotes both have cell wall, but the chemical nature of prokaryotic cell wall and eukaryotic cell wall is different. In prokaryotes, it is made up of a molecule which is called peptidoglycan. But in case of eukaryotes, it is made up of polysaccharides like cellulose. And prokaryotes, they divide by a division which is called binary fission, but eukaryotes divide by mitosis. Now the prokaryotic cell, a typical prokaryotic cell is the bacterial cell. Pro means first. Karyotic is from karyon. Karyon means nucleus. Means these are the organisms which have primitive or the first type of a nucleus, which is not bounded by any membrane or nuclear membrane. They just have a chromosome which is present in the nucleus in a specialized region that is called nucleoid. Just like eukaryotic cell, they also have plasma membrane. Outside the plasma membrane, there is cell wall. And outside cell wall, some of the bacterial cells, they have capsule. They also have extra nuclear material, that is plasmids, pili or pili, 70S ribosomes, flagellum, and cytoplasm. Now, the bacterial cell consists of cell envelope, which is called glycocalyx, and inside the glycocalyx is a cell wall. Then cell membrane, cytoplasm, nucleoid is the region where chromosome is present but not bounded by any nuclear membrane. And cell appendages, that is flagellum and pili or pili. Now the first one is cell envelope. Cell envelope, or the covering of the cell, is made up of glycocalyx and cell wall. Glycocalyx is of two types, and it is made up of glycoproteins and polysaccharides. And this glycocalyx, it surrounds or it covers. It is present outside the cell wall in some of the bacteria, not in all bacteria. As I told you, glycocalyx is of two types. One is called capsule and second is called slime. Capsule is tightly attached to the cell wall. It gives a sticky or gummy appearance to the bacteria, and capsule helps the bacteria to attach to the host tissues. But the slime is loosely attached to the cell wall, so it can be easily removed. It gives slippery appearance and rough appearance to the bacterial cell, but slime prevents the cell from phagocytosis or to be damaged by the phagocytosed cells of the immune system. And cell wall, that is second part of the cell envelope. It is made up of a complex or a large molecule that is called peptidoglycan or murine. What is the structure? What is peptidoglycan? We will do that in the chapter of prokaryotes. There are some bacterial cells which are without cell wall or they lack cell wall. These bacteria are called mycoplasma. This figure is showing the capsule and the slime. As you can see, this A1 is showing a slime which is green in color over here. You see it is loosely attached to the cell wall and can be easily removed, and it is rough in appearance. So it gives rough appearance to the cell or to the bacterial cell. And this prevents the cell to be phagocytosed. And this one is capsule. Capsule is tightly attached to the cell wall. It is smooth in appearance, so it gives a sticky or a gummy appearance to the bacterial cell. Inside the cell wall, there is a cell membrane. Cell membrane is just like the cell membrane of eukaryotic cell. It is made up of lipid bilayer, but the only difference is that cholesterol is absent in the cell membrane of prokaryotes or a prokaryotic cell or a bacterial cell. Remember, cholesterol molecules are present between the two lipid molecules at different places. So this cholesterol is absent in case of the cell membrane of a bacterial cell. Cell membrane in the bacterial cell at certain places invaginate to form foldings or infoldings, which are called mesosomes. As you can see in this figure, this yellow color infolding, which is invagination of the plasma membrane, this is called mesosome. It is believed that mesosome have some enzymes and it also have some functions to perform. It is involved in the transport of materials. It have enzymes for the cellular respiration, for photosynthesis, and also enzymes for DNA replication. So it is also involved in the division of the cell. Then inside the cell membrane, cytoplasm is present. This cytoplasm, as it is a part of a bacterial cell, so it is bacterial cytoplasm. This bacterial cytoplasm is different from the eukaryotic cytoplasm, as it lacks cytoskeleton, or cytoskeleton is absent, and the major difference is that membrane-bounded organelles are absent, except ribosomes, which are present in the bacterial cytoplasm, but these ribosomes are smaller as compared to the eukaryotic ribosomes. And just like the eukaryotic ribosomes, bacterial ribosomes are also made up of two subunits, larger subunit and smaller subunit. Larger subunit is of 50S and smaller subunit is of 30S. When they combine together, they make up 70S ribosomes of the bacterial cell. Cytoplasm also consists of some inclusions or some granules. These granules may have a molecule or a food molecule which is required to be stored, and usually it includes glycogen, proteins, and fats. Or these granules may have a waste in it which is required to be released out of the cell, and that waste is, or it may be alcohol, lactic acid, or acetic acid. Now the nucleoid. Nucleoid is actually a region where chromosome is present, or one single chromosome is present, but it is not bounded by any nuclear membrane. So it is the nuclear region of the bacterial cell, and it appears lighter as compared to the rest of the cytoplasm in electron microscope. As I told you, it consists of single double-stranded DNA. So this single double-stranded DNA is also called bacterial chromosome, and as one single chromosome is present, so the bacterial cells are haploid, and bacterial organism is a haploid organism. Haploid means half or one chromosome in it. And they lack introns. Intron is a part of a DNA which do not have a message for any protein or for any character. We will do that in detail in the coming chapters. And the nucleus, or this chromosome double-stranded DNA, or the bacterial chromosome, it controls growth and metabolic activities of the bacterial cell. In many bacterial cells, not in all cells, but in many bacterial cells, there is extra small circular double-stranded DNA, which is called plasmid. It is extra because it is present only in some of the organelles, not in other, and it is besides that circular double-stranded DNA or bacterial chromosome. It is believed that plasmid have genes which provide resistance against antibiotics, against drugs, and heavy metals. So plasmid have some extra genes which give some extra qualities or features to the bacterial cell. That feature is resistance against antibiotics, drugs, and heavy metals. This is a figure of a bacterial cell. You can see that here the outer covering is cut apart to show the outer capsule or glycocalyx, which may be a capsule or a slime. Inside the glycocalyx is a cell wall. Then inside the cell wall is a cell membrane. Cell membrane bounds the cytoplasm, which have number of ribosomes in it, and it also have some storage granules which are also called inclusions. It have this single nucleus and extranuclear material, which is in the form of a plasmid. And many bacterial cells, they have flagellum, which is used for locomotion. And many bacterial cells, not all, they have these small projections on the surface of the cell. These are called pili or fimbriae, and their function is for attachment. Now, what is flagella and pili or fimbriae? Flagellum are just like the flagellum of the eukaryotic cells. They are long thread-like structures used for the locomotion. But the difference is that in the eukaryotic cell, we have just seen that flagellum is made up of microtubules. But here in prokaryotic cell or in a bacterial cell, it is made up of a protein that is called flagellin. And pili or fimbriae are the tubular extensions made up of cell membrane which extend out on the outer surface of the bacterial cell. And pili are made up of a protein which is called pilin protein. Fimbriae are short as compared to pili, and their function is almost same. They are used for the attachment. But pili or fimbriae, they are present only in certain bacteria, not in all bacteria. Pili are present only in some gram-negative bacteria. Gram-negative and gram-positive are the two groups of bacteria classified on the basis of their staining. We will do that in the chapter of prokaryotes. And pili, as I told you, they are used for the attachment during a process of sexual reproduction that is called conjugation. Pili helps in the attachment of two bacterial cells, make up a cytoplasmic bridge. Through that bridge, genetic material is transported. In this figure, as you can see that this is one of the bacterial cell, which is named as F+ cell. This is second bacterial cell, which is named as F- cell. And pili or pili, it helps in the attachment of these two bacterial cells, so make a connection between the two bacterial cells. This connection is in the form of a cytoplasmic bridge, and the plasmid is present in one of the cell or in F+ cell but not present in F- cell. So one of the strand of the plasmid, it gets separates from the other strand, and through that cytoplasmic bridge, which is formed by the attachment of the pili, it moves to the second cell or to the adjacent cell. And both of these single strands of the plasmid in each cell, they will duplicate and make new plasmid in the cell. Now the bacterial cell division. Bacteria are rapidly growing organisms. They divide by binary fission. As you can see in this figure, when the cell is about to divide, what happens? The cell elongates and the chromosome, it duplicates itself. When the chromosome is duplicating, cell is keep on elongating or increasing in size. And as the chromosome duplicate, one chromosome moves to one side, other to the other side, and in the middle of these chromosomes a furrow appears which divides the cell into two daughter cells. This type of division is called binary fission. But the eukaryotic cells, they divide by mitosis and meiosis. So this is all about this chapter. I hope everything is clear and you people are understanding each and everything. Now in the end, there is an exercise. In this slide, I have given the solution of the first question, that is select the correct answer. Please, there are almost 15 statements and the correct answers are given over here in this slide as well. Your assignment is that there are short questions in the exercise from question number 2 to 25. You have to write down the answers of these questions. The answers should not exceed more than 4 or 5 lines, and the answers should be to the point, precise, and short. And these type of answers usually are the part of the section B of your paper. Thank you very much.